Good morning, children. In the last class, we were completed the topic until the sex determination, isn't it? Today, we will start the discussion with respect to the another important topic and wonderful topic that is nothing but mutation. Okay? So, before we are going to discuss about the mutation, let me to ask a few questions. Generally, you must have heard about. So, there is a two-headed cough. Okay, and sometimes abnormally, at the time of the birth only, one leg may be missing. Okay, so not only in case of the animals, sometimes in case of the humans also, you must have heard about the two-headed baby, isn't it? And sometimes, so there will be a additional organs may be appeared, or sometimes may be missing. Okay, so what may be happened all these in all these cases, so these are all it is because of the mutation. Okay, so in this class we will get the answer for all those questions. So that is nothing but wonderful topic. We'll start the discussion. That is nothing but mutation. How can you define the mutation? See here the few photographs it is given the left side. The baby, okay, with abnormal eyes and lips, and here the two-headed baby, isn't it? That's what I said. How it may be happens? What may be the reason? It is because of the mutation. When it comes to the definition, or how can you define the mutation? It is a sudden heritable change occurring in a DNA sequences that results changes in the genotype and the phenotype of an organism. Okay, so it can also be defined as, that means the mutation it can also be defined as a spontaneous and permanent change in the genetic makeup that can alter the characters of an individual is known as mutation. Okay, and the recombination and mutation, it leads to the variation in DNA. Okay, when it comes to the mutation, actually there are two types of the mutation, the positive and negative. If whatever the desirable characters appears in the next generation, then it will be positive. Instead of positive, if any negative mutation is appears or abnormals, then generally, in generally we can say it is a negative. Okay, see, according to the, that means Dobzhansky, it's the name of the scientist, okay, who is the first, the first person working about the mutation. According to the Dobzhansky, that means the mutation is a mistake or misprint during cell division. Okay, so I will repeat once again the name of the scientist, that is Dobzhansky, Dobzhansky. Okay, according to it is Dobzhansky, mutation is a mistake or misprint during cell division. Okay, and the mutation may be caused due to errors during DNA duplication or due to environmental factors like chemicals, radiation, exposures, unprotected exposures towards the dangerous radiation. That is also reasons for the or cause of the mutation. Okay, and if these mutant genes are in the somatic cells, they may not be of evolutionary significance. So that's what I said. If these mutant genes, that means the heritable changes, whatever it is happens, those mutant genes are in the somatic cells, they may not be of evolutionary significance. If they are in the germ cells, they are passed on to the next generations and are of evolutionary significance. Remember, so this is the next question generally. They will ask, when will be the mutation play very important role? during evolutionary significance. That too, if they are in the germ cells, they are passed on to the next generation and are of evolutionary significance. So, in generally, the evolutionary significance mutation is considered as the positive mutation. Okay, for example, the parents may be normal IQ, but their children will be very high IQ level. Okay, and generally sometimes the parent may be not that much of the height or mediums, the dwarf, but children they may become very tall with respect to the very good posture of the body. So then those will be considered as a positive evolutionary significance with respect to the mutation. And remember, in the beginning only I told you the mutations are considered as the raw materials for the evolution. If there will be no mutation, then there will be no evolution. Because of the mutation only, so generally whatever we are today, it is evolutionary characteristics. Whatever it is happens with respect to the plants, animals, including everything. So hence the mutations are considered as the raw materials for evolution. Okay, so you should note down all these things. Fine. I hope not only just listening while you are listening to this and watching this video, please note down all these points. Okay, yeah. And in your textbook, actually they have mentioned when it comes to the types of mutation, mutations are broadly classified into two types, that is point mutation and frame shift mutation. In some textbooks it is also mentioned three types. No problem. The point mutation is also known as gene mutation. Okay, so let us discuss one by one. In that one, the first one is a point mutation. The point mutation is also known as gene mutation. Okay, here only one example is given. Okay, it is a mutation due to the change in a single base pair of DNA. Example, we can give sickle cell anemia. Okay, so before going to explain about this point of gene mutation, just observe, consider the original sequence of a single base pairs. So here it is arranged like this, beginning from T A A C T G C A G G T. You know very well that what are all these things. The base pairs. T means thymine, A means adenine, and what about the C? Cytosine. G means guanine, isn't it? That is ITGC. Okay, but if you observe in case of the point mutation, so just observe what is the problem happened here. So that means TAAC. After that C, in original sequence, T should be there, but what happened here in point mutation, instead of T, C is came, isn't it? So that means when the mutation occurs at the gene level is called gene mutation, or at a particular point, that's why it is also known as point mutation. Okay, and so this point mutation happens due to change in the nucleotide sequences of a gene, that is nothing but DNA molecule. Okay, for example, in defective hemoglobin, the amino acid, as I said, we can also give in case of the sickle cell anemia. What may be the reason? Okay, note down this one. In defective hemoglobin, the amino acid glutamic acid is replaced by valine, where G, A, G codon is altered as G, U, G, codon on mRNA. Okay, then this results in genetic disorder, sickle cell anemia. Okay, so that's what they have given the the representation. The right side you can identify the DNA level TTC. Okay, then in the silent levels a nonsense, that means after the production of the particular protein it should be stopped. Then the conservative and non-conservative. But mRNA level generally normally they have given, instead of AAG, if it will comes AAA, and we can also identify the protein levels. Lysine AAG codon produces, but what will happen is, due to the point mutation, again AAA, it leads to the defective, that's a gene mutation. Okay, so that in case of the stop codon also you can identify UAG and AGG, that is arginine, and ACG thrombin. So this is how in case of the point mutations the defective genes is responsible for the mutations. It leads to the negative impacts on offspring. Okay, are you able to get it? Fine. The next one is frame shift mutation. The frame shift mutation, actually how it happens, the addition or deletions of base pairs of DNA, it causes frame shift mutation. Okay, so remember the frame shift mutation, how it will happen, it is the deletion, okay, or insertion of the base pairs. What will happen, sometimes a particular base pair completely it will be missing, that's what we call deletion. And sometimes insertion, newly the base pairs will be added. Instead of normal number, the more number, one or two more base pairs are added, it resulting in the shift of DNA sequences. Okay, so then what will happen, there are, as I said, the deletion or the gain, insertion or duplication of the DNA segment cause chromosomal abnormalities also. Okay, so then we can also call abbreviation, chromosomal aberrations. So the chromosomal aberrations are generally seen in cancer cells. Okay, so you know what is a cancer? It is uncontrolled mitotic cell division, isn't it? Due to the failure of, that is, protein synthesis, generally after the completion of the particular production of the protein, it should stop. But whenever there will be, due to the missing of nonsense codon, it will go on continuously, start repeating divisions. That's what we call generally cancer. Okay, yeah, the chromosomal aberrations are generally where we can see in cancer cells. Okay, yeah, so here the just observe they have given the point mutation versus the frame shift mutation, how it will be generally. If you just read as the particular frames, CAT, 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 CAT. Okay, so this should be a normal nucleotide sequences, so that it will produce normal protein synthesis or normal protein products. Okay, but second sequence is just observe, after CAT, instead of C, that means cytosine, additionally instead of C, G is get added. So that what will happen, the abnormal Okay, it is an abnormality with respect to the particular point. Then this will be reason for the abnormal but still functional protein products. So that abnormal protein synthesis will be takes place. Okay, yeah, finally what happened? So here generally whatever it will be to produce the proteins, they are abnormal. Okay, so this is how exactly we can identify the that is point mutation versus the frame shift. Are you able to get it? Yeah, in the third sequence CAT, after CAT is missing. Okay, yeah, so instead of now CAT, what happened? The CATC it is became. So that's what the complete frame, the addition or deletion of the base pairs of the DNA. So this is what we call the frame shift mutations. Okay, so the right side, for the purpose of understanding, so in the easiest manner they have given, consider in a normal case, if you take a spelling of the beast, B-E-A-S-T, beast. Okay, so in case of the substitutions, instead of B, if F new point, that means the codon, if it is get added, what will happen? Instead of beast, feast is happen. The complete meaning will changes, isn't it? At the same time, if you take an example of insertion also, after the B and before the E, if or is get added, the new point, that is nothing but the base pair is get added. What happens here? So that is nothing but breast. Okay, so then the deletion, so between B and ST, A is get deleted. So now what happens? It will become best. Okay, so at the same time we can also give inversion. After BA, if the S and T interchanged, then it will become the beads. So this is how exactly, okay, the generally we can give example for the frame shift mutation by comparing with the point mutation. Okay, yeah, and there is one more thing. We will discuss about the chromosomal mutation in this chapter on the last topic, but let me to tell you, in some textbooks and competitive they have mentioned the chromosomal mutations also. Note down, the chromosomal mutation, it is a kind of mutation caused by the change in the chromosomal number. It could be due to the addition or deletion of one or two chromosomes in the diploid set called aneuploidy. Okay, it's about about the chromosomal mutation. At the end of this chapter we will discuss once again. So that means addition of an extra X chromosome results in the Klinefelter syndrome, that is a feminine male, whereas the deletion of one X chromosome results in Turner syndrome. So then a sterile female. Okay, about the chromosomal mutation we will discuss once again in the at the end of this chapter. Okay, yeah. So after this one, as we were discussing about the mutation, isn't it? Now the question arises, how it will be, what is the reason for this mutation? The mutation is happens, as I said, the mutation can be defined as a spontaneous and permanent change in the genetic makeup that can alter the characters of an individual. But what is the reason for the mutation? The mutations happens because of mutagens. Okay, what are these mutagens? The mutagens are the agents that is reason or that induce mutation. Okay, or we can also say the agents that causes mutation is called mutagens. Okay, yes, the mutagens operate either by causing changes in the DNA of the genes or changes in the nucleotide sequence of the genes, so that interfering with the coding system or by causing the chromosome damage. Okay, so based on this, it can be divided into, that means the mutations are broadly divided into two types, that is nothing but types of mutagens: physical mutagens and chemical mutagens. How they are classified? The based on, that means how they interfering with the coding system or by causing the chromosome damage. That's what I said. The physical mutagens: ultraviolet radiations, okay, X-rays, UV rays. Generally, ultraviolet radiations are nothing but UV rays and X-rays, and also other shortwave radiations, isn't it? And alpha, beta, gamma rays. Generally, the person who is working with the ultraviolet exposures in the hospitals and all, okay, so then they undergo exposure of all these radiations without without taking any safety measures. They may suffer from this, the mutations. And chemical mutagens, generally the mustard gas generally uses in mustard gas. Generally, where it is used in the, yes, so cleaning, or you must have heard about the wherever the storing systems and all, there they will use this mustard gas, and phenol, formalins, acetic acids, formic acid, and also ammonia. Okay, so these chemical mutagens are just like, we can also give, you must have heard about EMS, ethyl methyl sulfonates, and also MMS, methyl methane sulfonates, and also colchicine. Colchicine also, they are considered as the chemical mutagens only. Okay, so these the mutagens are responsible for the mutation. Because of these mutagens, so generally the mutations happens. Okay, so that's why if the person who is working with this, the dangerous radiations as well as this chemical mutation, they should be careful. Okay, fine. So then after, the next topic is pedigree analysis. What is this pedigree analysis? You must have heard about the general terminology family tree, isn't it? So this is how they have given here the representation. It is an analysis of inheritance of traits or characters for several generations in a family. Is called the pedigree analysis. Okay, yeah. See, generally, what is important? What do you use? If you, generally, take an example of foreign countries. If they want to marry any persons, both persons, they undergo the complete checkup with respect to the pedigree analysis, so that they can identify what are the problems may arise in the future and what may be earlier happened. On the basis of that one, they can avoid the further problems. But especially the countries like India, in human, control crosses are not possible. So the study of the family history about the inheritance is generally used. Okay, such an analysis of genetic traits in several generations of a family is called pedigree analysis. Okay, and in the pedigree analysis, the inheritance of a particular trait is represented in a family tree over generations. Okay, that means from over generations to another generations. Okay, and as I said, in human genetics, the pedigree study provides a strong tool which is utilized to trace the inheritance of a specific trait or abnormality or diseases. Okay, the representation or chart, whatever it is given here showing the family history, is called family tree. So in generally we call it as a family tree. In genetics we call it pedigree. Okay, in human genetics, pedigree study is utilized to trace the inheritance of a specific trait, abnormality or diseases. So if they come to know in the earlier only, or if they are able to guess what might the problem arises in future, so that as much as possible they can avoid, isn't it? After the marriages and after the children, if they get the abnormal children, they cannot kill, isn't it? Instead of that one, before that only, if they are able to trace all those things, so that they can avoid maximum, isn't it? So here are the symbols. Generally we use these symbols. Some of the important standard symbols used in the pedigree analysis have been shown here. It is given the male or female, sex unspecified, the affected individuals, the mating and mating between relatives. That is consanguineous mating means the marriages between very nearest relatives, then those will be considered as the consanguineous matings. And if it will be given the symbol like this, the parents above and children's below. Okay, yes, and the parents generally with affected male child, it can be indicated like this. And 5 unaffected offspring. Whatever the number, if it is given in the inside the affected offspring, then it will be like that 5, 4, 3, whatever the number it will be given. If it will be 5, 5 unaffected offspring. If it will be 3, 3 unaffected offspring. Like that it will be given. Okay, yes. So here they have given an example for that is pedigree analysis helps to understand whether a trait is dominant or recessive. Okay, so generally, so there are two types of the traits we can identify, that is autosomal dominant trait pedigree, and another one is autosomal recessive trait pedigree. Okay, when it comes to the that is also autosomal dominant trait, we can also give one of the example, myotonic dystrophy. Okay, here the example how it will be. So just observe properly. This is the first, the case. In case of the autosomal dominant trait pedigree, the affected individuals have at least one affected parent. So you can identify here whatever the female, the affected female, when she marries with the normal male. Okay, so that's what it will be. Affected individuals have at least one affected parent. So the phenotype appears every generations, but unfortunately the reason is you don't think that all the children may become affected only. Unfortunately, half of the 50% of the possibilities, half of the children they may be lucky. and half of them may become deceased. Okay, yeah. The two unaffected parents only have unaffected offsprings. Okay, and so the traits are controlled by dominant genes. So both males and females are equally affected. Traits do not skip generations. So this is what it is given in the first case, that is autosomal dominant trait pedigree, that is myotonic dystrophy. You can also give another examples. Note down. Polydactyly. You must have observed there are some people, they will be having 6 fingers in the hands, okay, or 6 toes in the legs. That is polydactyly. Okay, so that is the example for the autosomal dominant trait pedigree. And you can also give another example, tongue rolling ability. Okay, so generally all the people, they cannot tongue roll completely. Only few members, they can roll complete tongue. So those tongues will also be very longest one. So those also example for autosomal dominant pedigree or dominant trait pedigree, tongue rolling ability. Okay, so this is with respect to that is autosomal dominant trait pedigree. Yeah, are you able to get it? Next one, second, note down, autosomal recessive trait pedigree. Okay, see here also, just note down few points. The unaffected parents can have affected offspring. In the first case, what happens? Affected individuals have at least one affected parent, isn't it? In the second case, it is very important that actually at the time of the marriage, both are normal. That means both male and female, the parents are normal. In this case, what happened? Unfortunately, because of the mutation, okay, unfortunately because of the mutation, the unaffected parents can have affected offsprings. Okay, so the traits controlled by recessive genes and appear only when they comes in the form of homozygous. This is neat question. Note down, the traits controlled by recessive genes and appears only when they comes in the form of homozygous. That is the best example for the autosomal recessive trait pedigree. Okay, and both male and females are equally affected after these cases. Okay, fine. Though the traits may skip generations, but remember, whatever the traits it skips, still sometimes they remains as in the form of the carriers. Okay, fine. And the neat question they can ask, it may be arranged in the form of 3:1 ratio between normal and affected is obtained. That means, see here only you can observe, after the parents there are 6 children are there. Out of 6 children, 2 children, one male and one female get affected. The remaining 4 are normal. So that means the ratio 3:1 ratio between normal and affected is obtained. Did you got it? Yeah, because for the purpose of the need, these points are important. That's why I'm telling. Okay. Thereafter, the appearance of the affected children form normal parents also in case of the heterozygous. The appearance of the affected children form normal parents in case of the heterozygous condition. But they may become autosomal recessive trait pedigree only in case of homozygous. Did you got it? Yes. All children of the affected parents are also affected. Okay. So that means if the person already is affected, if they marry, That means all the children of affected parents are surely they get affected in the next generations. So best examples for this autosomal recessive type pedigree, write down sickle anemia and albinism. Okay, in both the cases, if any one of the parent already if they are suffering from such a type of the disorder, surely in next generation, that means they will appear. So surely the diseased person will appear in the next generations also. Okay, about the sickle cell and all, I mean in the next coming classes I will tell you, because when we are discussing about the genetic disorder, I will tell you in detail about that one. Okay, yeah. So this is one of the analysis, the pedigree analysis. So this is also while we are studying about the hemophilia, I will tell you in the next class. But just observe, she is actually Queen Victoria was a carrier of hemophilia. So her family pedigree shows many hemophilic descendants. Okay, the normal female, the normal round symbol is a normal female, and it is a cube, normal male, off shaded carrier female, completely shaded cube, that is a hemophilic male. Here are the generations. Okay, that hemophilic descendants. The first generations, Edward VII, then the George V, George, that is the sixth, beginning from the Edward VII actually, George V, George VI, and Elizabeth II. Then the Alice, who she is actually a carrier female, isn't it? Are you able to identify? Then there is another example, that is Alicoplon. He is actually hemophilic male. In such cases, half of the children they may become safe. Then half of the film surely get affected. Then the carriers, in the next generation surely it will appear. The carriers are nothing but they look normal, but in the next generation surely it will appear. So these are all about the pedigree analysis. So let it be. Next class we will continue with respect to the genetic disorders. Thank you. Have a nice day.